To begin with our workshop series, let me take this opportunity to introduce you to our first speaker for the day, Dr. Tuhin Subrasantra. Sir is an assistant professor in the Department of Engineering Design at Indian Institute of Technology, Madras. Dr. Santra is a tenure track honorary visiting professor at National Tsinghua University, Taiwan, from 2018 to 2023, and he was a visiting professor at University of Cambridge, UK in 2019. A PhD holder in Bio-Nano Electromechanical Systems, BioNEMS, from National Tsinghua University, and a postdoctoral researcher at California Nanosystem Institute, University of California, Los Angeles, USA. Sir's main research areas are BioNEMS, MEMS, single cell technologies, single biomolecule detection, micro and nano fabrication, biomedical micro and nano devices, nanomedicine, etc. Currently, he is serving as a guest editor for Frontiers of Bioengineering and Biotechnology, Cell, MDPI, and Micro Machines journals. Having served as a guest editor for International Journal of Molecular Science in 2018, 17, 15, Census Journal 2016, Journal of Molecules 2016, etc. Sir also has been the editor of books like Handbook of Single Cell Technologies by Springer Nature 2021, Single Cell Analysis by MDPI Switzerland 2021, Microfluidics and BioNIMS Devices and Applications by Jenny Stanford Publisher USA 2020, Bionanomaterials by Springer Nature Publisher UK 2020, etc. Dr. Santa has been bestowed with many honors and awards such as Wellcome Trust and DBT India Alliance Fellowship in 2018, Honorary Research Fellow from National Tsinghua University, Taiwan in 2018, Bharat Vikas Award in 2017, IEEE NEMS Best Conference Paper Award in 2014, and a silver medal from Vidyasagar University in 2004, etc. Sir has published more than 45 international journals, has 20 US, Taiwan, and Indian pending patents, 8 books, 20 book chapters, 20 proceedings, and around 25 international conference papers in his research field. Today, Sir will be delivering the talk on the topic biomedical micro and nano devices for cellular therapy and analysis. We welcome you, sir, to deliver the talk. Over to you, sir.
Yeah, thank you very much, Astha, for your nice introduction. And thank you, Professor Iru, for your invitation to give a presentation in this talk.
Thank you, sir. It's our pleasure.
So today I will give this presentation the title about that biomedical micro nano devices for cellular therapy and diagnostics. And in last couple of years, we tried to develop a couple of devices for different intracellular drug delivery and for then further in vivo applications. So initially we tried to develop the device for in vitro application. So here mostly we develop couple of devices such as that nano electroporation device, photoporation device, micro electroporation device, different kind of photoporation device, so particular for cellular therapy and analysis. So mostly we use the concept for either electrical energy to break the cellular membrane for intracellular drug delivery, or we use that laser energy to break the membrane for intracellular drug delivery. However, currently we are working some mechanical stress-based intracellular drug delivery and other topics. Now, first, that important things is in this drug delivery. So delivery something into cells, why we need something that molecular delivery into the cells. So here, the cell engineering, where we can develop the different kind of device and different kind of molecules. So those are the like cell impermeable, mostly that molecules that we can deliver into the cell. So using this cell engineering technique, different kind of device which can precisely break the cell membrane or deform the cell membrane, and it can create the nanopores into the cell membrane, and then we can deliver that biomolecules into the cell. Then gene therapy, as you know, that specific gene we can just deliver into that inside the cell, and by that particular gene we can change the hormone inside the cells, or we can just replace that defective DNA inside the cell, and by that way we can immune the cell. Then cell imaging technique, where exactly particular some biomolecules we can deliver inside the cell, and then at that subcellular level we can just stain that particular cell, and then we can analyze different kind of for cellular analysis. So now the MEMS microelectromechanical system for cell, where nowadays a lot of MEMS device also developed for that cellular analysis. So this is just for example like intercellular pressure measurement for cellular deformation study, or even different kind of conductivity or permittivity measurement or capacitance measurement, impedance measurement. Nowadays you can use that different kind of that MEMS device. So now these are the popular drug delivery system, which is most common delivery system nowadays, and till it's have like lot of advantage. So first, the concept is that endocytosis process, where we have the different kind of biomolecules in just outside of the cell, and on timing that cell membrane can the bilayer lipid membrane, it can allow that very small molecules. However, the disadvantage of that, your molecule can enter throughout that membrane, and it is very time consuming and uncontrollable delivery. So even like if you want to deliver the DNA, even it can take more than 3 to 4 days, just specific DNA, and most of the DNA it is difficult to deliver by using this endocytosis process. Then gene therapy using virus, so particular like specific virus you need to bind that DNA or whatever gene, and then you can deliver inside the cell. However, the disadvantage is it's have like you need the particulars or specific virus, and the toxic issue is that main issue for that gene therapy using virus. The bulk electroporation for intracellular delivery, it is a most common concept, and where we have that cuvette, and that have like two electrodes, and the gap between two electrodes is 1 centimeter, and you can put the millions of cells together. And if you apply kilovolt or electrical voltage, then many of the cells can damage, and couple of the biomolecules can deliver inside the cell. So it cannot be controllable and cannot be precise or localized delivery. Then other common concept is microinjection technique, where people just put the needle inside the cell, and during withdraw this kind of needle, and you have that that membrane deformation, that large area of the membrane can damage, and this kind of technique is that cell can easily damage, and it is very hard to handle this kind of microinjection technique. So by using this common concept, it's have couple of disadvantage, and then so the most common question for this kind of intercellular drug delivery and therapy, that important question is, can you deliver that large cargo delivery inside the cell? The most of the device or most of the technique is failed to deliver different kind of large cargo in inside the cell. Then high delivery efficiency, of course, whatever the technique you are using, we need that kind of the delivery efficiency should be high. And most of this intracellular delivery, we need that after the delivery, your cells should be viable, and then we can analyze different kind of disease or cellular analysis. So if you deliver that cells and then deliver the molecules into inside the cell, and then it is just within couple of time, like it's just dead, that cells are dead, then it is useless for to do different kind of analysis. So intracellular delivery, like you need that delivery efficiency should be high, and your cell viability should be high after your delivery. Then delivery into cell directly, we can do this where we can perform this kind of directly biomolecular delivery inside the cell, and all cell type delivery, which is very difficult till nowadays. Even in one technique, it is almost impossible to deliver different kind of biomolecules in different type of cell. And then high throughput delivery is very important nowadays. So mostly people are moving for single cell analysis or single cell therapy, where we are almost familiar that compared to that bulk therapy or bulk analysis of the cell, if we can go to precisely just one by one cell analysis, so that can give like very precise data compared to that bulk therapy. So that's why single cell intracellular delivery using that high throughput delivery is very important for to do that single cell analysis, and after that you can just analyze cell to cell variation or cell to cell genomics or different kind of omics analysis. And then easy, we must need to like easy to handle that device or any kind of technique that is important. So in 2011 from MIT USA, that Professor N. Rusk, that he make one commentary article in Nature Method that is there any easy method to deliver that unpackaged cargo in different kind of cells, or even that large molecules in different kind of cells with high delivery efficiency and high cell viability. So this is the most important task for intercellular drug delivery, where we need to deliver that different kind of different size based cargo, and in different kind of cells we are able to deliver or not. And of course, this should be with high delivery efficiency and high cell viability. So coming to this point, then we tried to deliver different kind of, we try to develop the different kind of devices, and we tried to deliver that different kind of molecules in different kind of cells. So we'll just explain a couple of our developed device, how we can perform this kind of molecular delivery inside the cells for cellular analysis. So here this slide is presenting that bulk and single and localized single cell electroporation, where uh, generally you have that for bulk electroporation, you have that cuvette, and this cuvette you have two metal electrodes, and these metal electrodes, the distance between two metal electrodes, it can be uh, 0.5 millimeter to 2 centimeter. Then you have that millions of cells is in suspension inside that cuvette, and whatever you have that biomolecules, so that is just surrounded that cell. And after you apply that electric field in between these two metal electrodes, then that electric field should be in kilovolt order, very high electric field. Your application voltage will be very high voltage. And then this kind of electric field which can interact with that millions of cells together. And whenever this electric field can interact with that cell, then your cell permeability and cell conductivity will be different from inside of the cell to that outside of the cell, because you have that surrounded medium of this cell. So, and this cell membrane can act as a, it can consider as a dielectric material. So whenever you apply that electric field, then this membrane can act as a junction capacitance. And due to this junction capacitance, and you have that permeability difference in between inside cells and outside cell, then that membrane can deform. It can create that ion channel and ion pumps, and finally it can create that nanopore throughout that membrane. And then whatever you have that biomolecule, so it can diffuse inside the cell from outside to inside the cell. So this is the mechanism for electroporation, where we can transfer that millions of cells. However, this kind of delivery is not uniform, and even on that same electric field, you have that thousand and thousand cells can be dead, and many cells will be transfected. So during electric field, then membrane can just deform. It can create the nanopore, and then molecules can diffuse inside the cell because of the concentration gradient. And then after electric field, so molecules can successfully deliver inside the cell. And if you tune this electric field, then cell membrane can deform, or bilayer lipid membrane can deform, and that successful molecule can deliver for further analysis. However, if you have applied that very high electric field, then it's have the tendency to deform this kind of cell membrane permanently, and then your cell can easily die, and you cannot do that analysis. So after this bulk electroporation, it started around 1975, around 1980. So it is still now very popular for different kind of cellular therapy and different kind of disease analysis in vitro. And then in 2002, from UC Berkeley, they started by using this microfabrication and nanofabrication technology. They started that single cell electroporation technique, where in the single cell electroporation, by using microfabrication or microfabricated device, that electrode which is just placed surrounding that one single cell, where your single cell diameter is around 15 to 20 micrometer mostly. And whenever you just place this kind of electrode, and your cell is just inside of these two electrode, So now if you apply that voltage in between these two electrode, then your electric field can highly intense in between two electrode. And now you can see that that cell membrane can deform and it can create the nanopore, and whatever you have that biomolecules, so that can enter inside the cell. And after certain time, after withdraw the electric pulse, then your bilayer lipid membrane can deform or reseal, and then whatever you have that successful molecular delivery inside the cell. So after 2002, then mostly this kind of single cell electroporation, nowadays it's booming research for different kind of single cell analysis. So this concept first came, it's called like localized single cell nano electroporation, which that concept came first time in 2013, because the target is after your delivery of the molecules, your cells should be viable. So the concept is by using again this micro nanotechnology and microfabrication, where electrode is just in small gap, which can be in nanoscale gap in between two electrode, and then your cell can seated in top of the electrode. Then only the small part of the membrane, if you apply that electric field, that applied voltage, external voltage, then your electric field can highly concentrate in that small region, and membrane can deform in a very small region. And then whatever you have that outside molecules, so that can diffuse because of the concentration gradient from the molecules can diffuse from outside to the inside of the cell. So by using this technique, then we can increase the cell viability, because if you compare that bulk electroporation where your membrane is completely deformed surrounding the throughout that cell, even for single cell electroporation, we can see that membrane can deform throughout that cell membrane. So that's why it's a very high chance uh, after your electric field application, so cell can dead easily like with instant time. But if you use this kind of single cell nano electroporation, which can precisely deliver that biomolecule inside the cell, and after successful delivery, that cell can be live for a longer days for different kind of cellular analysis. So to using this concept, so first time we bring this concept, this localized single cell nano electroporation in 2013 from our group, and then how we develop this kind of single cell nano electroporation and micro electroporation, that I'm going to demonstrate that couple of device. So this is massively parallel high throughput single cell electroporation, where we applied that uniform electric field in each cells, and then we try to understand that that biomolecules is delivered into the cells uniformly or not. So here we have that silicon device, and in this silicon device, by using that microfabrication, we just fabricate this kind of thousand and thousand vertical channel, and this kind of channel diameter is around 15 micrometers. So it depends upon our cell dimension. So if we have that blood cell, then this dimension for red blood cell, like it is 8 to 10 micrometer, or for white blood cell, it can be 7 to 8 micrometer. So this experiment we tried for different cancer cell lines, so mostly HeLa cell and other suspension cell line, and we fabricated this kind of vertical channel is 15 to 18 micrometer. And in top we have that gold electrode, and now we just applied that external voltage. So here that positive polarity, and this is you have the negative polarity one by one. So whenever we just activate with that power supply with that external voltage, then in between two electrodes, so positive and negative polarity, then electric field can highly intense in between two electrode. So now the bottom of this device, we connected with that PDMS material, and which make the cavity, and then we connected this syringe on that PDMS, and it is connected with that syringe pump. So this syringe, it's contain that different kind of cells. So here just we considered like one cancer cell, and then it mixed with that cargo or biomolecules, and then by that syringe pump we just pressurize by that syringe pump these molecules, and which can go to this PDMS chamber, and then finally this kind of cells and molecules which travel from this kind of vertical, uh, the direction that cell can be travel through this vertical channel. So now whenever this cell can travel, and the cell dimension, it use like HeLa cell, and which dimension is approximate 15 to 17 micrometer diameter. And whenever it can passes through this vertical channel, then it can interact with the electric field in top side of this electrode or top side of this device. And whenever it can interact with that electric field, then its permittivity and conductivity is changing, and that's why it can deform the cell membrane and can create the nanopore. And then by diffusion process, whatever that molecules so that can diffuse inside the cell. So by using this device, so this is the first time we developed this kind of high throughput parallel single cell electroporation, where we can just in 2 to 3 minutes we can use this kind of 2 into 10 to the power cells per ml, and which can deliver that biomolecules uniformly, because we have that same electric field throughout this device, and whenever that cell is passing, so uh, interacting with that uniform electric field throughout this device. So now by using this device, so this is the device fabrication where we consider that silicon on insulator wafer. We have the 300 micrometer silicon, then silicon dioxide is 500 nanometer, then another 10 micrometer the device layer, and then both side we have that silicon dioxide layer, 1.2 micrometer. And then step two, like we use that photolithographic process, and which can pattern that photoresist on that bottom side, and then we use that reactive ion etching process to etch the silicon on that bottom layer. Then step three, we use that direct reactive ion etching technique, by we just press that particularly etch that silicon from this bottom side. And then we etch, after etching that silicon, then again we use that photoresist coating and use that reactive ion etching technique to etch that silicon layer. And then also in the top side we pattern by photolithographic process, that photoresist patterning. The next step is that gold deposition by e-beam evaporation to form that electrode pair. And then again that PR coating and hole formation using that direct reactive ion etching. And then finally we etch that silicon layer by the direct reactive ion etching. So this is the final hole formation using that direct reactive ion etching technique on silicon substrate. This is that scanning electron microscopy image where you can see that these two are the electrodes, and this is the gap between two electrodes, and this hole is around that 15 micrometer. So this is the top view where we can it is clearly visualized that we have that gold electrode, and uh, this is the hole is around 15 micrometer uniformly we created, and this is the side view after that direct reactive ion etching that 15 micrometer hole. So now by using that device, this device, so by syringe pump, we just push that cells from that bottom, and it can just go, or it can come into that top side of this device, and it can interact with that electric field, and it can deliver that molecules. So using this concept, and we also simulate that by COMSOL multiphysics, that electric field and resistive heat simulation. So here we can see that this is gold electrode, and this is the hole around like 15 micrometer hole. And whenever we can apply that around 10 volt uh, uh, applied external voltage, then we can see that that electric field, it is just surrounded that hole, and which can be nearly like 2.5 or 2.6 into 10 to the power 6 volt per meter, and which is high enough to deform the cell membrane and create the nanopore for delivery of that biomolecule inside the cell. And so this is the zoom where we can see that electric field just highly intense into that uh, surrounding that hole. And again, same thing, like we calculated the resistive heat at whenever we applied that 10 volt external voltage, then we can see that we have that gold electrode, and then mostly that resistive heating also, it is just edge of that hole where we have that gold electrode. So, and this kind of heating also is around like 8 into 10 to the power 11 watt per meter cube. So using this kind of electric field and heating, which can deform the cell membrane and deliver the molecules inside the cell. So now this is experimental results where we use that calcium molecules, which is cell impermeable molecules, and whenever it can deliver inside the cell, due to that membrane deformation, and then this kind of molecules can provide that green color inside the cell cytosol. And after delivery, then we use that PI dye. So PI dye generally it is cell impermeable dye, but if your cell is completely dead, then it can enter automatically inside the cell and it can stain the nucleus of the dead cell. So here uh, by electroporation we deform the cell membrane, and calcium molecules is delivered inside the cell, and red color is indicating that we have that dead cell after the delivery. So by this is the flow cytometry results where we can confirm that around 92% that calcium molecular dye is delivered inside the cell. And however, you can see that this is high throughput electroporation device. Even we apply that same uniform electric field, but this kind of human cervical cancer cell, it's deliver calcium molecules in most of the cells. However, there are a lot of cells and that are die even we applied the same electric field. So by these results, we can even do that cellular heterogeneity analysis. So even we are same stage of that cells, we applied that electric field, uh, but the most of the cells are molecular dead, that molecules are delivered inside the cell, but many cells also dead even that same electric field. So here we applied that kilohertz or a 10 kilohertz and different applied voltage 3, 4, 5, 6, 7, 8. And this is the percentage of that the delivery efficiency and cell viability. And we can see that around 4 volt 10 kilohertz pulse, uh, around 91 to 92% delivery efficiency, with the cell viability is around approximate like 82 to 83%. At whenever we applied the 10 megahertz pulse and with the different applied voltage, then we can see that on the 10 megahertz pulse, because you have that 10 megahertz, it's approximately like 100 nanosecond pulse. And then we can see that delivery efficiency is nearly around that 85 to 88%, where your cell viability is around 90%. Then we tested for different molecules. So dextron molecules, dextron 3000 molecules, and this is cell impermeable molecules. And whenever we applied that electric field, then cell membrane can deform and dextron molecules can deliver inside the cell and it can provide that green color. And after delivery, we added that PI dye. And this PI dye, whenever you have the dead cell, it can automatically stain the nucleus of the dead cell and it can indicate that dead cell. So here we also can see that dextron molecules successfully deliver, however, that many cells are dead. And whenever we use the dextron 70,000, that very, very large molecules, and then we can see that we have that syringe flow rate and we use that 10 volt 10 megahertz pulse, and we can see that to use very large molecule where that delivery efficiency is very less, and with that syringe flow rate is around like 1 ml per minute, and uh, the delivery efficiency is around like even less than 10% whenever we use this kind of very large molecules. So that means in this device, even we applied that high voltage and it is not enough to open the very large port. And so this device can be delivered like up to dextron 3000 molecular delivery successfully and with that very high delivery efficiency. However, very large molecules it is unable to deliver. Then we use that suspension cell for delivery, which is very important. Most of the device is unable to deliver that any suspension cell for intracellular delivery. So we use the THP1 suspension cell where plasmid delivery is successful and which can express that protein expression. And by flow cytometry result, it's almost approximately like 68% delivery efficiency we can achieve. Then another device which called the nano localized nano electroporation device, which is US patented. The previous device also US patented, and this device we just published in Lab on a Chip in 2020, where we have that bottom is that silicon substrate, and top we have then we have that silicon nitride layer, and top we have this is indium tin oxide, which is very transparent, and we fabricated that electrode by microfabrication nanofabrication process, and gap between two electrode is just 60 nanometer gap. And then we have a vertical channel in between two nanoelectrode tip. And in that bottom silicon substrate, we make that micro channel into that bottom where it is connected with that inlet channel, uh, and that channel is connected with this inlet and that outlet. So by syringe pump, we just whatever that cargo or biomolecules, we just push by this syringe pump so that cargo or biomolecules can go to that bottom of this device, and then it can flow through this channel, and outlet is sealed, and you have that vertical channel in between two tip. So that's why that molecule can continuously flow from this bottom channel, and it can continuously flow from that bottom to top of this device. So now we culture the cell, and whenever we culture the cell, and some cell are seeded in between tip of this nano electrode. So this is just a zoom image where that single cell is seeded uh, in in between two tip, and then if we applied that electric field, and then when we applied that voltage, then electric field can highly intense in between these two tip, and then due to this highly intense of that electric field, only a small region, then cell membrane can deform, and even very high electric field which can make a very large nanopore. And then whatever you have that biomolecule, so that we just push by syringe pump which can deliver from this bottom to that inside of that single cell. So now this work actually just by Hindu Business Line, they just that media news came on 2020 throughout India. Their scientific page, it's called that Quantum. And then we tried to estimate that electric field intensity. So here we can see that this is the nanoelectrode where gap is around like 60 nanometer. And if we applied that around 6 volt external voltage, then we can see that electric field can highly intense in between two tip. And this is parallel electrode. And in this device, we have around 30 parallel electrode, and each electrode can intense this kind of highly electric field, which is around 1 into 10 to the power 8 volt per meter, and which is high enough to deform the cell membrane and create the nanopore only in single position of that single cell. A resistive heat calculation, again, it is showing that the resistive heat also can highly intense in between two tip. So here that zoom image of the nano electrode 60 nanometer gap, and if we apply that 6 volt applied voltage, then we can see that heating also is affecting into that tip, and which is around 1.4 into 10 to the power 16 watt per meter cube. So here electric field and resistive heat affect together to deform the cell membrane, even that single cell, and only the single position we can make a big hole, and then biomolecules can deliver by that pushing that molecules by that syringe pump. So now the chip fabrication, I will not go to that very detail. So here this is the basic concept where single cell is seeded. Then we have that silicon material. Then we have that silicon nitride, which is transparent material. Then we have that silicon dioxide layer, which passivate that electric field in unwanted area. And we have that inlet where by that syringe pump, we just continuously deliver the biomolecules. And when we just seal the outlet, then this kind of biomolecules can forcefully, it can go through this kind of that nano channel, and then it can deliver inside the cell when we apply that electric field to deform that cell membrane. So now here, this is the basic fabrication. We consider the 225 micrometer silicon wafer, and both side is 100 micrometer, 100 nanometer silicon nitride layer. Then we etch that nitride etching. We did that photolithography pattern, and then we use that reactive ion etching to etch the nitride layer on that bottom. And after that, we use that potassium hydroxide to etch that. By using this kind of chemical etching, we etch that silicon layer until when it's remained that nearly 30 micrometer silicon. Then we did that oxide formation by thermal oxide. And after that, again, we pattern by photolithography, and we etch by reactive ion etching this oxide layer. And then we use that tetramethyl ammonium hydroxide, which can etch that silicon material that pending that remaining 30 micrometer silicon layer. So now we can see that only 100 nanometer suspension layer in top of the silicon substrate. Then we coat the ITO, which is top of the silicon nitride layer, that ITO indium tin oxide, which is also transparent. And to passivate the electric field in unwanted area, then we deposited that silicon dioxide layer. And after that, we use that focused ion beam etching to etch that or to make that vertical hole in between two electrodes. Uh, and we make that 60 nanometer gap in between two electrodes. And then finally, we did that glass bonding on bottom of this silicon wafer. So the reason is that the whole device is transparent because this is very friendly, and it's just 225 to 226 micrometer the thickness of this chip, and which is very compatible with that inverted fluorescence microscope because this chip is completely transparent, and we can visualize that intracellular drug delivery on top of the single cell by applying that electric field. So now this is chip fabrication, complete fabrication, where we can see that this is that nitride membrane, which is around 400 into 400 micrometer suspension membrane and 100 nanometer thickness of the membrane. And after that, we just pattern by photolithographic process, and this is ITO lines. And this is the ACM image where we can see that these all are that indium tin oxide ITO lines. And after that, we etch by focused ion beam, and we make this kind of very tiny triangular shape nanoelectrode formation. And just one triangular shape nanoelectrode, we can see here that 60 nanometer gap between ITO nanoelectrode. And now we have the whole surrounding that nanoelectrode. And whenever we can push the biomolecules from that bottom of this device, so that biomolecules or DNA which can come from that bottom to the top of this device in this area throughout this hole, and your cell is seeded in this area, and by electric field cell membrane can deform in this area, and then whatever that larger biomolecules can deliver inside the single cell. So this is the chip fabrication, and then final device fabrication. This is the final device where we have that inlet and outlet, and around 30 to 35 that nanoelectrode pair into this chamber, where we culture the cancer cell or stem cell. And then on that specific position, when cell seeded into the top of the electrode, then we apply that voltage for cellular deformation and for nanoelectroporation purpose. So this is the experimental results. Again, here PI dye is cell impermeable dye, but if you deform the cell membrane, then particular that deformation, that PI dye can enter inside the cell because you create the nanopore, and whenever you can create the nanopore, then PI dye can enter inside the cell and it can stain into that whole cytosol. But if your cell is dead, then automatically your bilayer lipid membrane is completely deformed, and it can automatically enter PI dye, and it only can stain the nucleus of the dead cell. So here you can see that this is our nanoelectrode, and where we have 60 nanometer gap, and this cell is seeded in top of the nanoelectrode. This is the CL10 human lung cancer cell. We applied that 6-volt peak to peak 20 millisecond pulse. And whenever we applied and surrounded, we have that PI dye, then we can see that very fine, precise molecular delivery. And after applied that external voltage and then electric field are highly intense, then this is that 1 second after applied that external voltage. This is 3 seconds. And then we can see that particular this area of the membrane will deform. So that's why PI dye is started to accumulate particular this area of the membrane, where we just on that single cell we just deform into that single area of the membrane, which is nearly 60 to 100 nanometer, and PI dye is entering slowly inside that cell. This is after 3 seconds of that visualization. Then we can see that after 7 seconds, 10 seconds, 30 seconds, 17 seconds. So around 20 seconds to 25 seconds, we can see that dye is completely diffuse inside the cell, and it is completely stained into that whole cytosol of this cellular inside of the cell. So now this is the real-time fluorescence microscopy video, because this chip, the beauty of this chip, like this is completely transparent of the chip, and by using that inverted microscope, we can track that molecular delivery inside the cell. So here we can see that we have that we applied that voltage in here 16 nanometer, and you have that single cell. And then whenever we applied that voltage, and only the membrane can deform in a very small area of that cell membrane. And then PI dye is completely diffuse after from this area, like where we make that big hole into that single cell membrane in nanometer scale area. And then molecule is diffused from this area, and like within it depends upon that applied voltage. So this is 5 volt 10 millisecond pulse for human colon cancer cell, and which can just deliver that molecules within 5 to 7 seconds inside that whole single cell. So then different molecular dye with the different pulse. As we know that if we apply that millisecond pulse, then your pore diameter will be that larger uh, compared to that microsecond pulse. So microsecond pulse or nanosecond pulse, if you apply, then your pore diameter will be the smaller. So your diffusion of this molecule will be slowly diffusion compared to that millisecond pulse. However, if you use that larger external voltage, then your affected area of the cell membrane will be that higher. So that is almost well developed concept about that electroporation. So here we use that microsecond pulse, and now we can see that this is the electrode position, and cell is seated just top of this electrode. And when we applied this kind of colon cancer, human colon cancer cell 5 volt 500 microsecond pulse, then you can see this is 10 second. Around 13 seconds, you can see that uh, that membrane is deformed and molecules or PI dye started to accumulate into that center position. Then slowly it is diffusing surrounded that cell. Then we can see that 80 second, 120 second, 150 second, around 180 to 200 seconds. And we can see that PI dye slowly diffuse from this only one position of the single cell, and then it is completely fully diffused inside the cell. So now then we use our device where we have the nanoelectrode gap between two nanoelectrode is 4 micrometer. But whenever your cell is seeded into top of this electrode, so cell is generally after seeding it is around 25 to 30 micrometer. Then we can see that two nanoelectrode position where we applied that external voltage, and then by the applied external voltage, electric field can highly intense in only two small position. Then we can see that if this is that human AGS, that human gastric cancer cell, 5 volt 300 microsecond pulse, and we can see 10 second, 20 second. We can see that this two position is confirming that uh, that hole is created only in single cell two position, and then molecule is started to diffuse from outside to inside the cell from this two position. And you can see after 20 second, 40 second, 50 second, 60 second, and around 90 to 100 second, that molecule is slowly diffuse inside the cell. So this is also well proved. Whenever we are going to that millisecond pulse, we can see that for real time video, it is like 5 seconds, within 5 seconds, that molecule can completely diffuse inside the cell. However, for microsecond, this is 500 microsecond pulse, which is taking around 200 second to molecular delivery. And if we use that multiple number of electrode, and which is taking around like 300 microsecond, is taking around like 100 second to 140 seconds to diffuse the molecule inside the cell. So this is just intensity profile of that cell. We just captured that intensity within time. So this is like after 30 seconds that intensity profile, and this is the cell dimension. And we can see that around 120 second, then because that molecule is delivering only the center position of this cell, because the membrane is deformed only the center position. So that intensity profile also confirming that your intensity is very high, because this position we deform the membrane and molecule is delivering inside the cell. So same thing here, whenever we use that multiple number electrode, two electrodes, then we can see that two position that intensity is high and molecule is delivering inside the cell. But whenever after certain time around 180 seconds, then your intensity is almost constant throughout the single cell, and your intensity is here we can see that there is no peak, and molecule is successfully delivered, and intensity is that constant throughout the cell. Then quantum dot delivery, again we use these two electrodes, and we deform the cell membrane in two position. And we can see again on timing, because this quantum dot is dimension is around like 10 nanometer, the large quantum dot, and it can take in minutes after the deformation. And we can see that around 28 30 minutes, and around that 40 minutes, that quantum dot successfully deliver inside the cell and it's stain as a red color. So to confirm this quantum dot delivery, then we use that confocal scanning image where we just scan from top to bottom and 1 micrometer scanning step. This is the top image and this is the bottom image, which is also confirmed that 1 micrometer step size for each cases. And it's also confirmed that quantum dot successfully deliver inside the cell. And your cell dimension is here that this is 1 micrometer step size, which have that 16 images, and cell dimension it confirmed that it's around like 15 to like 17 micrometer cell, which quantum dot successfully deliver inside the cell. Then we use that large plasmid. This is colon cancer cell. We applied 6-volt peak to peak 20 millisecond and 3 pulses together, where you can see that this is 3 nanoelectrode and 1 single cell. And we just deform that 3 position of the membrane. And then we visualize that GFP protein expression. So generally, if you put by endocytosis process, most of the DNA cannot deliver. Even some of the DNA can deliver, but it can express that protein even after 3 days. By using this technique, you can see that this is the hottest which can stain the nucleus of the cell with the different time, plasma membrane staining, dye. And then after that plasmid delivery, that this is the GFP expression, that important results where within 2 hours, we can see that protein is expressing into that cytosol. And we can see that after 6 hours, even after within 8 hours, then that protein can express into that single cell. So then this is the Mars image of the nucleus and membrane. And this is the membrane and GFP protein expression. And this is the nucleus and GFP. As we know that protein is expressed just cytosol, and which is also confirmed that you have the nucleus of the cell and that protein is expressing into that cytosol of the single cell. So now we'll go to that another technique where we use that another concept where we have that substrate, glass substrate, and we culture the cell, and then we design some specific nanoparticle which have that asymmetry kind of peak and which can enhance that electromagnetic field, and then it can deform the cell membrane. So whenever you have this kind of very special design of that nanoparticle which is attached into that cell membrane, then we expose that pulse laser, and due to exposure of that pulse laser, this kind of gold nanoparticle which can intense that electromagnetic field and surrounding medium, and due to that intense of that electromagnetic field, and this kind of gold nanoparticle can heat up, which can produce that nano bubble surrounded that gold nanoparticle, and this nano bubble can force the membrane and it can deform the membrane. And then if you have that biomolecules which can outside of that biomolecules, so due to deformation of the membrane, it can deliver inside the cell. So this we just published in RSC Nanoscale in 2020, this concept. And where this kind of special design particle, how we fabricated that silicon substrate, oxygen plasma treated substrate, and then we fabricated that PDMS oil, and then we dropped that polystyrene beads into this PDMS oil. So after a certain time, this kind of that polystyrene beads which can rearrange as a hexagonal structure. And after this kind of hexagonal arrangement, then we use that reactive ion etching to etch this kind of polystyrene beads. And after etching, then we coated gold on top of this polystyrene beads. So this is by e-beam evaporation, that gold deposition, and we fabricate that special nanoparticles. So this is the we call that mushroom shape gold nanoparticle, where you can see that there is very sharp peak, and this gold nanoparticle is nearly 300 nanometer, and it's have more than 60 to 70 that peak, like which are very sharp 30 nanometer peak into that gold nanoparticle. So if we expose that laser and this kind of peak, which can act as an optical hotspot and which can enhance that high electromagnetic field compared to that normal spherical particle. And then this kind of particle can easily deliver that molecules into the cells with very low laser fluence. So now, then this is the human cancer cell, lung cancer cell, and then we seeded that particle, this kind of very special design nanoparticle into that lung cancer cell. And then we tested that UV spectroscopy, which is around, you have the near infrared absorbance spectroscopy is around 945, and we have around like 530 nanometer 2 absorption peak. Then we use this kind of 940 nanometer peak for that laser to intense that laser to expose that laser on particular this kind of near infrared region. And by that exposing the laser at this region, then this kind of particle which can create that nanobubble, and that nanobubble can force the membrane or break the membrane, and then molecule can deliver inside the cell. So this is we just use that COMSOL multiphysics simulation where we can see that we compare with that spherical shape gold nanoparticles and a mushroom shape whatever our special design nanoparticle which have very tiny peak. And we can see that the field enhancement is in spherical nanoparticle. We can see nearly that 60 wherever this kind of very special design particle which can enhance around nearly 300 that magnitude that field can enhance. And then the distribution of that temperature distribution of that particle, we also can see that this is the temperature distribution which is nearly same. So here by inducing this kind of electromagnetic field as well as heat which can deform the cell membrane and successfully deliver the molecules. So this is the different kind of cell we use, that human lung cancer cell, human gastric cancer cell, and P19 stem cell, which is very difficult to transfect the cell using any of the physical technique or any of the chemical based process for intercellular delivery. So we use very small laser fluence, which is 30 millijoule per centimeter square. And we put that PI dye surrounding that cells, and then we can see that PI dye successfully deliver and it can stain the nucleus. It can stain the cytosol. And PI dye, if can stain only the nucleus, then it is confirming that it is dead cell. So here, this is the cytosol staining. Then we use that calcine AM, which is cell permeable molecules, and it can stain the whole cytosol. It can produce that green color, which can confirm that your cell is live and completely cell cytosol staining. So by these two images, we can confirm that your cell is live and PI molecule is successfully delivered inside the cell. And this is the Mars image, where also confirming that successful molecular delivery at near infrared wavelength, that we use the 10 Hz pulse for 30-second exposure. And in this Mars image, we can see that P19 stem cell, couple of cells are dead, which is because we marked that red and green color. So we are expecting it is green to yellow is green, but those are like completely red color, which confirm that cells are dead and it is staining that nucleus of the dead cell. So then we again use that quantum dot. Again, we can see that that human lung cancer cell, gastric cancer cell, and P19 stem cell, that it already stain and successful delivered that quantum dot, confirming this red color, and that cells are live by using this viability test. Then control experiment for plasmid delivery and protein expression. We can see that different kind of plasmid, which is 3500 BP and 4779 BP, which we base pair, like we expose that laser, and then we can, it's also confirmed that that plasmid can successfully deliver and protein can express after 24 hours, particular those area we expose by that laser. Using this nanoparticle, we also that tested first time in the world that 3D scanner ferroid based delivery, where we make that 3D scanner ferroid and we mix this nanoparticle and we expose that laser using this nanoparticle around like 680 nanometer with this special design nanoparticle. Then we successfully delivered that different kind of molecules into that 3D scanner ferroid, and which is published in which is patented and also published in RSC Analyst. And this work is, due to the novelty of this work, is accepted as an analyst cover page of this invention. Then another device, which is again that photoporation based device, and we use again this is the silicon material which have that hollow vertical hole, and in top side we culture the cell, and we have this kind of very array of 3 micrometer holes. And in these 3 micrometer holes, we just coated the titanium into that surrounding the holes, and then we expose the laser. So whenever we coated this kind of titanium, and when we expose the laser, this titanium can heat up, it can form that cavitation bubble, and this cavitation bubble can force the membrane and it can break the membrane, and then we deliver the large molecules from bottom to inside of the cell. So This work is commercialized, so I will not go to the in detail due to the limitation of the time. This is the fabrication process we use where this device size, uh, this is just like 1 centimeter square is the device size. And by using this concept, then whenever we expose the laser, then we form that cavitation bubble with the different laser exposure. And we can see around 35 to 50 millijoule, which like a cavitation bubble can form surrounded that titanium deposition. So here we tested that calcium delivery. So this calcium is cell impermeable, and whenever that membrane can break, this calcium can deliver inside the cell and it can produce that green color. And after delivery, we use that PI dye. This PI dye, if you have that cell membrane is completely cell membrane is deformed and cell is dead, it can stain the nucleus of the dead cell. So by this delivery result, we can confirm that nearly 45 millijoule to 55 millijoule per centimeter square, where that most of the cells that calcium can deliver and a couple of cells are dead, which is indicating by that red color. However, if we increase that energy, then cells are dead, more cells are dead. So by using this technique, we deliver that bacteria, very large bacteria, 2 micrometer. So this is that membrane opening time, and this green color is confirming that bacterial delivery inside the cell. So this is like the actin staining on that. We use the different kind of bacteria and into that cell cytosol. This is the actin staining into that cell. Then another device, it is accepted in SCS Biomaterial Science and Engineering, which is patented. The concept is we fabricated that array of titanium disc. It's 3 micrometer titanium disc, which is 100 nanometer thickness. And then we culture the cells in top of the device. And then we expose or scan the area. So this device is around like 2 centimeters square. And whenever we scan the area, then titanium can heat up and it can produce that cavitation bubble, which can force the membrane and deform the membrane, and then molecules can deliver inside the cell. The device fabrication, that complete microfabrication process, we use that device fabrication. Then we deliver into that different kind of cells. So this is again that PI delivery into uh, human cervical cancer cell. This is that neuron cell. This is again human cervical cancer cell with 21 millijoule per centimeter square energy. And then the viability is confirming by that calcium AM dye. And then we use that different kind of other molecules to confirm this device efficiency. We use that dextran delivery, we use that siRNA delivery, and different kind of DNA delivery by using this device. Then this is the last device where we did that single cell printing technology and intercellular delivery, where we considered that glass substrate. And then we use that SU8 we coated on top of the glass substrate. And then we pattern this SU8 and we make the thorough hole. This hole dimension is around 40 micrometer to 80 micrometer array of hole. Then we release this SU8 membrane. And after SU8 membrane release, this membrane we just put into that Petri dish and we culture the cell. And after 15 to 20 hours, then we release this kind of SU8 membrane. And now we can sedate this kind of array of cells. So our target is to do this kind of cell to cell signaling or single cell analysis or cell to cell variation, cell to cell genomic study. So here we did that intercellular delivery study. So after this kind of patterning of the cell, then we added that biomolecules into the cell. And then we use that this kind of array of gold coated disc, and we put into this glass substrate this kind of gold coated disc, which we put in top of the cell, and we expose. We did the laser scanning. So by this laser scanning, this kind of gold disc, it can heat up and it can produce that cavitation bubble, which can deform the membrane, and it can deliver that biomolecules into that array of cell, which is seeded in throughout this kind of area, which is 1 centimeter square area. So using this concept, we do the single cell therapy, and this is the fabricated device and ACM image of this array of hole. And then this is the experimental results where we use that 40 micrometer SU8 hole, and which we can see that a single cell to two cells can print 45 micrometers holes. That cell is printing, and then we stained by that calcinum, which is confirming that live cell staining. And this is 50 micrometer SO8 hole, which is printed that branch of cells together. So this is also confirming that 60 micrometer, 70 and 80 micrometer different kind of cell printing for single cell analysis and that cell to cell communication study, even that cancer cell to that normal cell interaction that we are doing that research nowadays. And also as well as we are doing that intercellular delivery. So this is experimental results for intercellular delivery. Again, more than 20 cells per pattern, less than 20 cells. So this is PI delivery using that complete device, and less than 10 cells patterning and less than 4 cells patterning successful deliver PI molecules. Then we the after delivery that all cells, almost 100% cells are live by confirming that calcinum staining of that live cell, and this is the Mars image of that cells. The conclusion is like we successfully, whatever our concern, that 3D electroporation device, that high throughput delivery we perform. So whatever the concern is large molecular delivery, we deliver that bacteria, different kind of quantum dot and different kind of plasmids. And high delivery efficiency, we achieved almost 98%, and some of the particular cell type even we can achieve 100% delivery efficiency with that 100% viability. And this is like directly cargo delivery into that cytosol. So whatever the concern, that easy delivery and easy handle or high delivery efficiency, high cell viability, we are trying our best to achieve this parameter for intercellular delivery. So thanks. So I'm the editor, and currently we publish this kind of related research work in Handbook of Single Cell Technology. I'm the editor. Nanomaterials and biomedical applications, microfluidic biomems, all are 2020, 2021, single cell analysis. Then currently I'm the journal guest editor, mostly for single cell analysis in Journal of Cells and Journal of Micromachines. So we applied like 2020 10 patents, and 2022 is 7 patents in this research field. And thank you for your attention.
Hello. Hello.
Yes, yes.
Yeah. Do we have any questions, or anybody wants to discuss anything with sir?
Thank you, sir, for your wonderful question, talk, and we are glad you are here. You have discussed a lot of information, the research work, which is mostly how we can do the some drug delivery and drug screening applications, and also you have discussed about like a lot of things like single cell suspension, how we can make. So yeah. Yeah, I would like to ask you if you guys have any questions. You can ask to the professors, to him, sir, and please you can unmute yourself and ask any questions if you have. Sir, from my end, actually, I have a couple of questions. Of course, the when we do some kind of the culturing on a device, so what are the precautions we supposed to take, when single cells culturing on a either it is a microfluidic device or the whatever the you prepared on the glass wafer as well as any other devices.
Yeah, the first is toxicity is the big issue. So we need to concern about the toxicity, because many of the microfluidic device have a lot of toxicity issue. And
Okay.
cell culture is going that healthy cell because of the toxicity that we are very concerned about that first. So we tune that different kind of materials and to reduce the toxicity. And after that we perform that this kind of single cell printing, and single cell printing also not easy task. And because of the printing, this kind of the hole should be uniform, and your that whatever the membrane should be attached into that petri dish very uniformly. So these couple of parameters we need to care about, and then finally we can successfully print the single cell by using this kind of SU8 membrane.
Okay, sir.
And also
One
we
more thing.
sterilize that
Yeah.
UV light, and sometimes we sterilize with that different kind of protein of that membrane, and then we use that membrane.
Okay, sir. One more thing, sir, like when we are like the culturing cells on top of these devices, do we need like the specific environment that so that only these like the cells will grow, otherwise they cannot, and how their shape will like changes in case of like if we don't have the proper environment there.
Yes, actually, we also fail many times to print it, because we are working last 4 5 years for this printing, and the result we achieved just in last 1 year. So that's I'm telling, like, your membrane uniformity is very important. That if that membrane is little bit of bending of that membrane, then cell cannot print. And also that the another important theme I told, like membrane should be attached into that petri dish. So
Okay.
sometimes that membrane is floating on that petri dish because of you have that cell culture medium. So that is
Okay.
also, it's not printing that cells properly. So this kind of fine tuning we need, and then successfully we can print the cell.
Okay, sir. So ideally, like what is the like the thickness of this membrane, sir?
It just now it is around like 10 micrometer, 10 to 15 micrometer.
Okay, okay, sir. Thank you, sir. And the Professor Sudeep Kundu would like to ask a question, I think.
Yes.
Yeah, good morning, sir.
Yes.
For your extensive this kind of one presentation, so it's nice presentation. And even though I am not at all related in the field of this bio nano part, I am from electronics background. So I have one query that whatever the devices you fabricated, uh, have you fabricated, is it in IIT Madras itself, or
Your voice is breaking.
hello, am I audible, sir? Hello?
Yes, your voice was breaking. Can you repeat the question?
Yeah. The question is, whatever the devices you fabricated, like the techniques you use, that DRI or RI, PECVD, all these are available at IIT Madras, or was it
No,
Kolhapur?
that mostly DRI, RI actually we used in abroad. Sometimes we use in IIT Madras also. We have available that RI and DRI, and sometimes we use IIT Madras. Sometimes we use in IISc. So
Okay.
just combine, like wherever the facility we are getting. Mostly we use IISc, IIT Madras, and in abroad, mostly in Japan and Taiwan.
So those are from by paying the fees, means.
Yes. It's too expensive process. Yes.
Yeah, sir, actually, I worked one energy harvester for these biomedical implanted devices.
Yes.
But due to this fabrication cost, we could not go for this fabrication. But we worked on this some piezoelectric energy harvester for pacemaker and so on.
Yes. Yeah,
Okay,
actually
so
it's too expensive, and we are also spending almost like 30 lakhs consumables per year nowadays.
yeah,
Every
that
year
30
almost 30
lakhs,
lakhs. Yeah.
that 30 lakhs even apart from this abroad fabrication.
Yes.
Correct.
Yes. It's too
Sir,
expensive.
yeah, can you just say, means how much cost this fabrication? Because we tried at soy mum's process and it was around 5 6 lakhs. So approximate what is your experience, that where is it possible to fabricate with a reasonable price? Uh, some basic sensors, some basic devices.
Yeah, like if I can see your device layer, then I can tell the estimated price. Like what kind of layer you are using, what kind of material you are using, then I can estimate the price. because
Okay,
each
okay.
material cost is different. Each process cost
Yes,
is
yes,
different.
absolutely, absolutely, sir. Means it was normal silicon and zinc oxide and just two metal electrodes. The basic device was, of course, some back etching and all those was there. Some, yeah,
Okay.
it is
Yeah. So
not
then it will
possible
be
directly.
cost will be reduced. But if you use that photolithographic process for patterning, then cost will be little high. So even in our laboratory, actually, we set up that all photolithography process and everything. We make the clean room in our own laboratory in last 3 years.
Okay,
And
okay, thank you,
mostly
sir.
the
So,
all equipment
yes,
we purchase from Taiwan with less cost actually.
okay. And is it possible to do some collaborative research? Means collaborative means if we get some facility for fabrication, those are available at national level, these equipments, or these are particularly
Yes,
for IT?
yes, yes, yes. Like I'm accessing that IISc. Actually, you can use that facility, but you
Yeah,
just need to pay the cost.
yes,
Yeah.
I am the member of this IEES and I tried at IISc Bangalore, but
Mm.
incidentally that time it was in 2019. That time that fabrication was little bit 1 year we could not get that facility. So
Okay.
otherwise, is it possible at your side also for this?
Yes, like for simple device, maybe I can help to you. Because now our group is actually biggest biomedical group in IIT Madras. We have now almost 13, 14 PhD student. We are the biggest group now
Okay,
in
okay,
IIT Madras.
okay,
So
okay.
our lab is too crowded actually. But if you
Yeah,
have
yeah.
couple of simple device and maybe will take little time, but we can do that.
Okay, okay. So for that we can contact directly you. Will it be in that way or?
Yes, yes. So we can do that. Some simple device we can fabricate, but maybe the time commitment maybe we cannot
No, no.
tell
Okay,
like
okay. I
within
am.
3 months we can deliver or something.
Yeah, yeah. I
Because like
understand.
equipment also have the many times equipment have problem, and in this pandemic also like everything is going to stop.
Yes, sir. Yes, sir.
But in metro city always COVID and always like the lab is going to close, and last 2 years actually work is not going so smooth.
Okay, okay. Yeah.
Yeah.
Thank you,
So
sir.
please
Thank you
contact
for
me.
your. Yeah, sure.
Yeah, please contact me, and if you have that simple device, then I can help to you. No, no issue. Yeah.
Okay. Thank you again, sir, for your so nice and extensive lecture. Thank you
Yeah.
also.
Thank you.
Thank you.
Thank you, sir. And I think, sir, IIC is like the costing around the like the for simple devices and other things. It is around 20,000. Suppose if you have the 4-inch mask, if it
Mm.
is the silicon wafer, then it is around close to 20,000. Like what I know. Okay,
Yes,
so
actually, it depends upon your process step. Like suppose
yes.
if you are just, I'm giving example in IIT Madras, I'm just using e-beam evaporation one time for
Mhm.
titanium deposition. They are taking 25,000 for one time.
Okay,
So it
okay,
depends
okay, sir.
on your equipment use and lithography. Like lithography now we have the all set up in our own laboratory, but previously I use IIT Madras facility. They use like per hour, it is like 2,000 to 3,000.
Oh, okay, sir. Okay. Yeah, in IIC also like the if we are in the like the faculties within the IIC, actually there the cost is different and the outside, even though we belongs to the institute, then there is a different cost, sir. So yeah,
IIC actually, there is one program that INUP. So your student can first join in their training INUP program. And
okay.
if you join on that program, then you can access that IIC facility with less cost.
Oh, okay, sir.
So actually, one of my student who was doing the PhD under myself, he did that INUP program, and then only we went for that, tried for that fabrication. Even then, the cost was approximately they estimated 1 lakhs per device. So even we could not support that using that. I was having that ACRB project, but that fabrication facility means support was not that financial support.
Yes, yes, I am
So
agree. So that's the problem of our country. Even we
even
cannot afford even IIT Madras. So it's
yeah.
difficult. Like nowadays, the funding is very difficult, and even many of professor like very good academic record. They are in IIT, NIT, even small IIT. Most of them cannot afford because of the funding issue and facility. I understood.
Yes,
So
sir,
that's
that
why
is
I'm
the
telling, like, if
problem.
you have anything simple device, I can help to you guys. No issue.
Yes, thank you,
But
thank you very
because
much,
we
sir.
have our own lithography process. All PDMS, PMMA, and all silicon-based device fabrication, we have our own laboratory. I
Okay,
build up
okay,
everything myself.
okay,
So like
thank
what is
you,
the
sir.
difference we have, sir, in the fabrication process, like the PDMS and PMMA? So PDMS, I'm aware about like the complete fabrication process, but the PMMA, like the how different it is with PDMS.
Uh, PMMA generally, so same thing, like whenever you have that SU8 material, then generally nowadays, like we develop a lot of PDMS device. PMMA, we just started it, and we till we are not using that PMMA actually. PMMA, we just use that laser patterning on that PMMA material. We just purchase from outside. But we are
Okay.
planning to use that PMMA. But PDMS, we did lot. So we have all the facility
Okay,
for
sir.
PDMS and for facility for silicon. We have so many, we purchase a lot of wafer also. So if you have that simple
Okay.
process, then I can help it. Even
Okay,
for
sir.
that mask, like because I have so many good contact in Taiwan and Japan, we are accessing all the mask from them. So
Okay, sir.
even the mask also here, cost is different. One mask like IIT is telling 1 lakh, same mask that ISC is telling 20,000. Even 20,000, they are delivering that mask after 6 months.
Oh,
So
okay, okay,
it's
sir.
like have a lot of problem, and whatever I have the good contact in mostly in abroad, Taiwan, Japan, I'm just purchasing from them, or I have a lot of friend, and they are actually freely delivering that all materials to me. We have like very
Oh,
strong collaboration. So
that's
if
Sir,
you
I
great,
have,
have
sir.
another.
So
yeah, simple device, then maybe I can help. Whatever we have the facility, I can help.
Sir, I have another question, just not related to your field, related to
Yeah,
somewhat to
sorry,
my field.
actually, yeah, your time is going on, so I
Okay, okay,
tried
okay.
to.
Thank you,
Yeah.
thank you, thank you.
No, no, no. You can talk because that Professor Iru, like maybe you have the another talk right in 11 o'clock.
yes, sir. Yes, sir. It is
Okay, no
actually
problem.
11:30,
Thank you, sir.
11:30, sir.
Oh,
So
they
no
okay.
problem.
Then you can.
We
Okay,
can
then
have fruitful discussion. There is no
okay.
issue, sir.
Then go.
Okay,
Yeah, carry
so
on. Yeah.
sir, one question is that from I am from electronics background, mainly on these signal conditioning circuits and designing of this for biomedical signals
Yeah.
or energy power conditioning circuit for energy harvester design. So can you say means but specifically what type of electronics challenges you are facing, means if something is there it would be nice for you, not related to optical side, not like fluorescent microscope, not like that, but from electronic side, means specific detection techniques. Is there anything like that?
Uh, yes, actually, I need to know like what kind of design you want to. You are planning to what kind of design and fabrication. That is the first important thing. So we are also working screen printed electrode for sensor application. Uh, so different kind of sensor we are working. So here, this is just one part we are doing the delivery. The another part we are preparing that microfluidics nanomaterial synthesis. We are working on periodontal with two doctors in Chennai. We are working in CMC Vellore with that other disease problem, and we are working also sensor development. So screen printed electronic device we are fabricating, and we are using that for saliva detection and uh, for different kind of that mostly that biosensor we are working on. So I need to know like what kind of design you want to do. And the other important thing, until we'll start the fabrication, actually you cannot understand.
Okay, thank you very much, sir, for your
Yeah, so
clarification.
that is
Thank
the most important
Sir,
thing. So whenever we are going
you.
to
Yeah,
fabrication, then we can understand that what kind of problem is arising. So
yes, sir.
each device have like each like different kind of problem during fabrication process.
Okay, okay, thank you, sir.
So we are working also for
sir,
glucose detection, different kind of. Yes, yes. Go ahead.
sir, I have one question.
Yes.
What is the difference between lithography and colloidal lithography?
Soft lithography and
Soft lithography. Colloidal lithography.
Your voice is not clear. Soft lithography and
Colloidal lithography.
collagen lithography.
Soft and colloidal lithography.
Colloidal lithography. I never heard that word.
Yeah, I too. I too never heard, sir. It is actually
Soft lithography is fine. Soft lithography is
soft lithography we know, and additive manufacturing we know,
yeah,
and
everything.
like stereo lithography also we know.
Yeah,
But
but what
colloidal
you are telling
lithography,
that I never heard actually. Sorry.
yeah.
Soft lithography means you are just using like that PDMS material to replicate the structure. So first you are making that SU8 based structure on silicon material, silicon wafer, and then whatever you are replicating from that SU8 mold or other mold. So this is just replication process. Generally, we call that is the soft lithography process.
Sir, it is possible by soft lithography to positively and negatively replication of
Yes,
mold.
yes, yes. So because your SU8 material is actually negative lithographic process, and but the structure whatever you want, so that can be fabricated. It can be possible
Okay, thank you,
by
sir.
soft lithographic process.
Okay, if you have any questions like the more, then maybe you can email to sir. So sir might be like he might be happy to answer you all. And thank you, sir. We had a
Yeah,
great
yeah,
session, and it was like the discussion also very fruitful.
yeah.
And
Yeah. Thank you. And sorry, actually, last 10 minutes I just speed up because as I know, like the talk is maybe 50 minutes and then one like 10 minutes discussion.
so actually, wantedly, I kept like the 30 minutes for like the gaps, sir, so that the fruitful discussions can happen. So 1 hour talk and around 30 minutes gap I kept, sir, actually.
Yeah,
So,
actually that
and
I was not familiar actually. So I was thinking 50 minutes talk and 10 minutes discussion. So that's why last 15 minutes I just speed up and just finish that off. Okay.
so for future collaboration and other things, I might be contacting you. Thank you very much, sir, for
Yeah,
your
yeah.
wonderful talk and
Yes.
everything.
Okay. Thank you, Professor Idu. Yeah. Thank you
Okay,
very
sir.
much.
Thank you,
Yeah.
sir. Yeah.
Thank you.
Yes,
Yeah.
Asta, you can conclude the meeting now.
Yeah. Thank you, sir, for delivering this highly informative lecture. We all are immensely grateful to you, sir, for providing us with the insights which would add to whatever little we know and whatever we plan ahead in this field of interest.